Okay, brilliant. Thank you very much. Thank you very much to Jamie and Anna for inviting me here today. It's a brilliant opportunity to showcase our facility at Leicester. So the Leicester hosts the Midlands Regional CryoEM Facility. It is a world-class state-of-the-art facility, and we provide the whole range of cryo-EM sample preparation to data processing. So the initial grant was set up in 2017 as a collaboration with our Midlands partners, Warwick, Birmingham, Nottingham and Leicester. And then in recent years, we've expanded to include Aston and Kiel as well. So we're really happy that we are expanding and continuing to grow our user community. We are a small but sweet facility, so Claudia and myself manage and work within the facility on a day-to-day basis. We work really, really closely with TJ Reagan, who is the CryoEM Computational Facility Manager, and the academic lead for our facility is John Schwabe. So John led the bid initially in 2017 to bring cutting-edge cryo-EM to the Midlands. We sit very firmly within the Leicester Institute for Structural and Chemical Biology, or LISC-B, and it's a really brilliant place to work. It's a collegiate environment. We have nearly 30 active research groups from multidisciplinary research, from chemistry, physics, biology, all sorts of interesting biological methods. And we're really proud of our facilities. Obviously, the best one is the cryo-EM, but there is also brilliant NMR facilities, new single molecule equipment, including C-Trap, and all sorts of different things. And we are super proud of the doctoral training program. So all of our PhD students come out as well-rounded interdisciplinary students who have knowledge of lots and lots of different techniques. And over recent times, we've been really trying to expand our international networks with a relationship with Lund in Sweden for links. And we have very strong regional networks, particularly for the Midlands. So a lot of our users do come from LISC-B, but we have a wide user base as well. So you can see that on this pie chart. So we've got Leicester and our partner universities, and then we work with academics such as yourselves across the UK and the world. And then we have a nice bit of industry usage as well. So the facility hosts sort of three main sections for cryo-EM. So we have the sample preparation equipment. Our VitraBot is the one that's used the most, but we have a manual single-sided plunger. And we very recently bought a SpeedBot from Steam Munich at the University of Leeds for time-resolved EM, and we are starting to see some really nice results from that. And we also have a whole host of everything that you need to get started in cryo-EM. We have a G3 Cryos with a Gatan K3 BioQuantum, and it's in a really beautifully stable room, so we get really nice data. And one of the things I think is a bit more unique to Leicester is we have a bespoke computational cluster built and designed by TJ, and this has GPU nodes, storage for data processing, has a queuing system, and everything is sort of set up ready to go in terms of processing software, rely on CryoSpark, and anything you might need. And another new addition is a dedicated Tomo PC. Although I should say that the majority of what we do is still single particle, we're starting to do more and more cryo-ET as well. And it's not just the equipment we provide, but we really try and make a whole sort of start to end training program for everybody. And we have expert guides and advice. And if you are located at one of the Midlands universities, this is free training, and that includes a session on the Cryos, where lots of people get some lovely preliminary data to go into grant. So if you're from a Midlands university and you don't already access us, come and have a chat with me. So our philosophy is cryo-EM for everybody. So what we need from you is your sample in a tube, and you don't need anything else. So it's really good for new users who don't have any equipment and don't know where to start. So we take you from sample in a tube to your beautiful cryo-EM figure, and then we sort of help you in the middle, and that really depends with what you need. Some people already know what they're doing; they're experts, but there's some of you out there that may not know where to start at all, and so we provide everything you need in terms of training for that. So that's hands-on training led by us, information on where to go and get the best training guides or online resources, but then we don't sort of just leave you to it either. We sort of help you every step of the way through the data processing as well. So we have online resources and we have a lovely community, and this is our weekly CryoEM club meeting, which is in person, and then we have a weekly data processing club run by TJ where you can ask any sort of simple data processing question or really, really technical advanced data processing questions. So it's really great to learn the processing side of EM. So we work on a variety of different things. This is an absolutely giant phage that we've been working on recently. And you can see a 78 kilodalton protein just in the can you probably just about see it, just to show you the range of stuff that we work on. But there's a huge sort of interest in DNA, complex proteins involved in DNA, replication, repair, all sorts of different things. So I'm going to tell you a bit from the Schwabe group. They work on HDACs or histone deacetylase complexes. These are really important in essential development and cellular processes. The HDACs are combined into multi-protein repressor complexes, and here's a schematic of 5 of them. And the Schwabe group work on them using a variety of different techniques, including a variety of different structural techniques. So I'm just going to show you 3 cryo-EM examples from Liam, Ed, and Louise. And so these structures you can see here, we still have some low resolution HDAC complexes, but the really brilliant thing about the Schwabe group is they're still able to get quite a lot of information from these low resolution maps, and you can see year on year the development that they've gone on in a lot of these complexes. So Louise has just published this lovely MIDAC work, and I want to tell you a little bit more about SMART. So Ed Brown, so hopefully most of you have bumped into Ed. He presented a poster yesterday. He's a brilliant postdoc in the Schwabe group, and he's been working on the SMART complex. So I think John's lab have been working on this complex for many, many years. And so when Ed joined, this was the cryo-EM structure that this is the structural information we had on SMART. And so he started afresh and tried to design a new construct for SMART. One of the main things that he did was he removed the WD40 domain. So I don't know if you've worked on proteins with this, but it really dominates all of the cryo-EM alignments. So he removed that from the construct. And also some flexible regions. Stuck it in AlphaFold 2, and it got 2 different structure predictions. And so we decided to use cryo-EM to try and find out which one of these models was, in fact, the correct one. And I haven't got lots of information on the grid optimization, but everything that you needed to try, Ed needed to try with this complex. It wasn't a whack it on the grid and it works. He had to go through loads of different optimization steps, cross-linking, addition of different inhibitors and things like that. But he managed to get it to work, and he's got these really lovely 2D class averages shown here, just to give you an example of the internal features that we can start to see. There was still some heterogeneity in this mix, so you can see the full complex here, and this is approximately a half of the complex. So there was still a bit of a processing challenge ahead of Ed with this. So about a year ago, this is where he was with the data processing. So he went through a fairly standard data processing pipeline and ended up with about 150,000 particles to get this structure. And it wasn't the best. He wasn't able to get loads of information from it. It had pretty bad preferential orientation, and it was over sharpened. And so one of the reasons I wanted to share this particular story is when Ed brought this to CCP-EM last year, he got some really good suggestions from chatting with this brilliant community. And it's actually changed the structure a lot. So thanks everyone for getting involved with that. So one of the things was Ed wasn't happy with the particles, the number of particles that he had. So, or that everything was being picked. And so some of the advice was, well, why don't you just use multiple pickers and combine them together? So he did that and then ended up with about 11 million particles. And then he did this particle seeding technique. So this was also presented at CCP-EM last year by Amano, and it worked well for Ed. So if you haven't heard of it before, you get your good particles. So this is all particles from the same data set. So he's got 150-ish thousand good particles. And then he's got his new 10.7 million particles from the template and blob picker. And then he splits these into different groups. So each group is seeded with 150 good particles. And then about 2/3 of the particle subgroup has about 300,000 of the new unknown particle stack. He takes those through heterogeneous refinement, and this is a multi-reference one, so or decoy processing. So we use multi-references, one good, so that initial map that you saw at the beginning, and then 4 bad maps of junk, essentially. And then take the good class into the next step. So that's one round of particle seeding, and he took it through 5 different rounds of that. And then in the end, ending up with 520,000 particles for further processing. And obviously quite important to remove any duplicate particles at each round. So we took this new data set, which is obviously quite a bit bigger than his initial 150,000 particles, and run it through homogeneous refinement. And the map was, the resolution was reported was, I think, the same. The map looks a little bit better, but you can see now that the distribution of views is much improved. And so we could take this into 3D classification without alignment, which was another recommendation from somebody at CCP-EM. And he could take the best class from that through homogeneous refinement, non-uniform refinement, and he got what turned out to be a really, really nice structure. So this is the overall map from Indeed EM Hansa. And from this map, he was able to see inositol phosphate that was co-purified with the complex, and also build up a story about what's happening at the tetramer interface, and a story about how HDAC and TBL1 interact together. So hopefully, if any of you want to find out more about that, you can speak to Ed, who I'm sure will be very happy to have a chat. And then I'll just move on to another story that we are in the process of finishing at the moment. So this is from Patricia's group. So Patricia is a chemist, and she's working on sort of green chemistry in helping to target net zero by 2050. So how can we lower those emissions? So one of the ways to do that is design efficient catalysts, for chemical energy conversion reactions. So Patricia's lab consists of herself, and then we've got Manon, who's done a lot of this work, and Aisha, who was just a master's student who's just finished. But with the facility, we were quite interested in this as research project. So Claudia and TJ have been heavily involved in the processing of this data as well. We've also relied on some friends from around the country. So both James and Jamie have Vitrobots in anaerobic glove boxes, which have been important. So I'll talk about that in a sec. So what we want to do, or what Patricia wants to do, is structurally analyse metalloenzymes. So this is turning small molecules into high-value chemical feedstocks. And currently in industry, this requires a lot of energy. But in nature, proteins such as CO dehydrogenases do this with very little energy. And CO dehydrogenases process the reversible reaction of carbon dioxide to carbon monoxide, which we can't seem to reproduce this in industry. So Some members, well, one member of this family structure has been studied by crystallography, and that's shown here. And we do have some information about how the enzyme works. This is the active site, and it's a 4-iron sulfur cluster. It's a bit different to the usual 4-iron sulfur clusters because it has this split, and one of the ions is replaced with a nickel. The exact mechanism of how this works isn't fully understood, and other members of the CODH family have increased rates of catalysis, but we can't understand why. So we really want to understand what's going on there. So the CODH enzymes that we work on, or Patricia's group work on, are highly oxygen sensitive. They come from thermophilic bacteria from volcanic springs. So they have evolved without oxygen, and the presence of any oxygen completely degrades these clusters. So to resolve any of the catalytic information, we had to produce the grids in the absence of oxygen. In fact, the whole protein production and purification happens in anaerobically, so it's quite a difficult protein to study, I'm sure you can imagine. It's 140 kilodaltons, and this particular CODH cannot be crystallized. So we've got lots of questions about it for it to be interesting for us. This is James's setup down at Essex. So James Burrell has Vitrobot in this chamber, and Jamie's is in a slightly different type of anaerobic chamber, but the same, obviously, principle. They're quite challenging to make grids in here, I'm sure you can imagine. It's quite a fiddly process, and you've got to wear these giant gloves. Also, when you add liquid nitrogen into the chamber, you do get some oxygen addition, so it's really important to keep an eye on the oxygen levels. Another thing that's quite tricky is the pressure in there. So when you're trying to apply the sample to the grid, the solution sort of pops back up the pipette as you're trying to get it onto the grid. So it's an extra challenge required to make decent grids. But Manon has been down to James's facility to make the latest batch of grids. And so she's getting a feel to how to do that well. So the initial cryo-EM studies were, we thought looked pretty promising from the micrographs, but had absolutely horrific preferential orientation that we really couldn't fix with any computational analysis. So we went back to the grid preparation to do some optimization and tried, I think, 10 or 12 different conditions with different detergents, different concentrations, that sort of thing, loaded up the cryos with the grids, and used multi-grid overnight to collect sort of 1,000 micrographs on each one of those conditions. So they're quite a standard screening procedure. And we just used our standard parameters. So they're just written on the screen for you there, but quite a wide defocus range. But this is just basically our standard settings that we start with and then fine tune to the protein. But what we found is this protein actually behaved really well in the presence of detergent, and we got a 2.2 angstrom structure from those 1,000 micrographs, which we were pretty pleased with considering how awful the initial processing was. And hopefully you can see the really nice density here for that. It is heterogeneous though, so this is the model I've just shown you, but quite a large percentage of the data has a really flexible lobe here, and it's difficult to resolve sort of 50% of one of these monomers. And this could explain why it was unable to be crystallized in the past. We did have a look a bit more at those iron sulfur clusters. So just to orientate you, this is the structure you've already seen, and we have the D cluster, which sort of links the two monomers together. We have two B clusters and then the C clusters, which are the active sites. So this structure is just turned on its side, and we can see where the clusters are again here. And they're responsible for electron transport, basically down from the exterior of the protein down through to the C site. So the D cluster, we saw really nice electron density for that. So this protein structure was just modelled with model Angelo, and then we docked in the ligands, and that's just turned on the side as well. So really nice resolution already. But unfortunately, for the sort of specifics in the, as you saw with the Rubisco talk yesterday, for looking at electron transfer, we do want super high resolution, really. But this is still a nice start. The B clusters, again, were really, really nice. And as you would expect, when you turn the contour level quite high, the protein sort of disappears, but those high atomic number ions still exist. The active site, however, wasn't as clear cut. So we do have nice density, but then when we do a high contour, most of the active site disappears. This particular sample, when we were doing the optimization, wasn't prepared anaerobically. So we're hoping that the oxygen sensitivity might have had some contribution to this. But we're really keen to see how far we can push the resolution to see if we can see those, see a lot more detail. And so in a very similar story to the Rubisco, we collaborated with Thermo Fisher and Simona's collected some data for us. We went back to the bench and prepared fresh sample, trying to push the boat out to try and get the best sample as possible, anaerobic grid types, hexafoil, and we had to use a detergent to sort out the preferential orientation. And these are the parameters that Simona's used on the Cryos 5. So pretty standard hexafoil settings and close to defocus. We did use a high dose, and the reason behind this was we were still unsure whether we would need to tilt the grid or not due to the preferential orientation, but preliminary data meant that we could collect the data flat, but we still went ahead using a high dose. So we got about 16,000 micrographs anaerobically, and then we also had an aerobic sample to compare it to, and this is an example of a couple of the micrographs. And then the initial processing that we've done so far, so unfortunately we haven't been all the way through the processing pipeline. I think TJ sent me this map on Tuesday night, is at 1.7 angstrom resolution, and this is sort of a very standard data processing. This is showing really lovely density. We can see that the hole in the histidine here, and we've got, again, really nice density for the D cluster, B cluster, and C cluster. And we've already got some ideas about how we can push that processing further. So watch this space. I'm hoping we can pull that down to much closer to 1.5 soon. But that's the end of that story for today. The final thing to finish off with you is animals in 2D classes. So I'm sure most of you have seen this. This is RNA contaminant, histag contaminants, and it's a beautiful dog face. And I constantly say to people, "Oh, you've got an animal in your 2D class average." So the people started sending me them. So we have a few examples from the Leicester community here. Lovely little bat or smart from Ed up there. And so I wanted to open this up to you guys, because I'm sure we've got some more animals in 2D classes in the audience. So please send your examples to me and I'll pop them on LinkedIn, and we can find the best one from that. So you can just email us at cryoem@le.ac.uk. So I'll just finish with saying thank you to everyone at Leicester and our community, our Midlands facility team and TJ and Claudia, and all of these people who've been involved in the work that I showed you as going through. Thank you.